Buenas tardes. No se escucha nada, ¿eh?
¿Escuchás ahora?
Ahora sí escucha. Gracias.
Genial. Sí.
Bueno, la reserva, mi Charla preguntó.
Sí. No, tiene que ser de la sala.
Porque conocer a algunas personas conectadas.
Sí, sí. Bueno, yo sigo tu paso.
Bueno, buenas tardes a todos y muchísimas gracias. Mi nombre es Fernanda Alonso, yo trabajo en el laboratorio Roche, la empresa farmacéutica, trabajo en el área de acceso y políticas públicas. Y es un gusto para ya estar hoy acá y hablemos alto, no hay problema, mirá que puedo hablar bastante alto. Entonces, bueno, buenas tardes a todos. Un gusto, Fernanda Alonso del laboratorio en Roche, trabajo en el área de acceso y políticas públicas para lo que es Roche Uruguay. Agradecer particularmente a David y a Teresiva por el espacio, también al equipo Uruguay 21 que nos ha ayudado a generar este vínculo. Y nada, es un placer en realidad para nosotros poder presentarles hoy a Magnus Fontes. Magnus es doctor en matemática, ha trabajado en bioinformática, modelos matemáticos, y es un matemático que se ha vuelto prácticamente un biólogo. Entonces es una gran convergencia de los mundos. Magnus trabajó en la Universidad de Lund, también fue director de un departamento de lo que es la red internacional de Instituto Pasteur, y desde esa experiencia ha tenido mucho contacto con todas las actividades del Instituto Pasteur en el mundo, y particularmente en Uruguay. Y desde hace, bueno, luego Head de Systems Immunology en Genentech. Genentech es Roche en Estados Unidos, y desde hace 3 años gerente general del Instituto Roche, basado en París. Y bueno, eso tiene su explicación concreta, que Magnus es la persona ideal como gerente general para contarles. Así que sin más, y si hay algo que falte de la presentación, Magnus, que es un poco informal, todo tuyo, y muchas gracias a la gente que está conectada también.
Muchas gracias, Fernanda, y sobre todo muchas gracias a ustedes de recibirnos y de darme esa oportunidad de presentar un poco. Y en realidad voy a hacer la presentación en inglés porque va a ser mucha, sobre todo ciencia, y me facilita mucho si lo puedo hacer en inglés. Pero ningún problema si hay preguntas, porque lo quiero tener más o menos informal. No creo que hay tanta gente online que entonces yo creo que podemos tener preguntas durante la charla, o si no, después. Voy a hablar más o menos 40 minutos y bueno, lo hago en inglés. Cualquier pregunta, ponela nomás. So, I guess you recognize this. Me imagino que ustedes saben dónde es eso. Eso es, ven, pan de azúcar está un poquito ahí a la izquierda. Es Tiriápolis, y el sol se está levantando. Eso es un mundo, y claro, hay un montón de otros mundos para mí, y voy a explicar un poco. So, I will explain a little bit my trajectory. So I studied mathematics, exactly as Fernanda said, and I did my PhD at Lund University. So this is the biggest university in Sweden. It's in the south. It's opposite Copenhagen, so it's just it's really a 30-minute train ride to cross from the Swedish side to the Danish side. So these cities are fairly close. So I did my PhD in pure mathematics. So I did it in, let's say it was harmonica analysis together with nonlinear partial differential equations. And I worked with Fields medalist, Lars Mander. But at the same time, I always did computation. So I'm fairly old. So when I started doing computation, this was in the late 60s, 1960s. I worked with a slide rule, see? And then 72, my father gave me a Hewlett Packard, that handheld thing. I could write 15 lines of code. And it was amazing, right? And then I got, 1980, I got the advanced basic computer for the 1980s, ABC-80. It was only produced in Sweden. I don't think it was extremely successful because nothing else happened, but it had 60 kilobytes of memory, 16 kilobytes of memory, and I could do anything, right? It was amazing. And then I had a series of other things when I started doing serious computing. And this actually led to the fact that when I was doing my thesis, I started computing, and in the late 90s, 1990s, this was after I did a postdoc in France, and I came back, and I actually became head of the department in Lund. So I was head of the Center for Mathematical Sciences for many years. But on the side, I was doing software for biologists. So I had a lot of friends. I think Lund is a mini Uruguay, actually. Everybody knows each other. It's a wonderful city. You must come there if you're in Europe, because it's you see, it's 75,000 inhabitants, 75,000 inhabitants. There are 50,000 students, 10,000 professors, and some kids, right? So when you go out on the street and you say, you don't ask what you do, what you work with. They say, what's your line of research? That is, how you start. And this makes a very, interactive and multidisciplinary field. And I think that in some sense, Uruguay can be I often see it as an expanded, much bigger and richer version of this mini, Lund feeling. So I will come back to this, but in any case, so I started working with medical doctors, and when the Affymetrix microarrays came, you could read 25,000 genes at the same time. You could understand what they were expressing in terms of mRNA at the same time. It was like, wow! And we did it for tens or hundreds of patients. And we had, Excel sheets, 7,000 pages of printed Excel sheets that they were looking at and trying to make sense of. Of course, it's impossible, right? Unless you know exactly what you're looking for. So then I started to do, principal component analysis, looking at things and doing clustering and classification. And that was very appreciated at that time. It was not a lot of people that were doing it. And that actually, after some years, I realized that, it doesn't really count for a professor that should be doing nonlinear PDEs to be, computing and coding for biologists all day. So then I started a company, Flucor, together with some colleagues. So I had a friend that worked as head of Ericsson Mobile Platforms in China. So I invited him to come. I had another friend, a medical doctor. I invited him. I had a brilliant friend that worked as software developer in the US, and I invited him. And together, the four of us started this company, Stuco. And actually, that company in some way led. I never worked for the company. I just helped to get it started, and I'm actually still on the board of the company. But I really never worked for the company. But it got me even more into contact with the bigger world of biomedicine. And that led to contacts with Pasteur Institute, first in Paris. And then I was actually recruited to head the bioinformatics and biostatistics for the global network of the Pasteur. So Pasteur is, you might know, I mean, you know very well Instituto Pasteur de Montevideo. And I got to learn to know them back in 2013, 14, more or less, when I started to work with a global network. 33 institutes in 26 countries. And actually that in turn led to other contacts. I also was working a bit in Copenhagen University. So for a couple of years, I was a professor there and helping out with bioinformatics, both at the hospital, Rigshospitalet, and also at the university. But then actually I moved over to Genentech. So Genentech is, as Fernanda pointed out, it's 100% owned by Roche. So internally in the Roche ecosystem, we talk about PRED and GRED. And PRED is basically all the research and early development in Europe. And GRED is Genentech. Here you see the campus of Genentech here, and then actually 3 years ago, again, as Fernanda said, I got the opportunity to join Institut Roche with headquarters in Paris. And actually, I mean, there are exactly two one-dimensional topological manifolds, either the circle or the line, right? So these are the two versions. So I went circle around and actually have now an adjunct professorship again at Lund University. So we kind of closed a little bit the loop. But that is, of course, just an adjunct professorship, which is gives me an opportunity to be in contact with and have PhD students. I think for me that maybe the biggest advantage. I still love teaching and so on. Roche is big. We celebrated 125 years last year. I must say that what convinced me to move from academia, like Institut Pasteur and Copenhagen University or Lund University, and move over to Genentech, which is part of the Roche ecosystem, is that it's the number one investor in research and development in the biomedical space. And we are ramping up our efforts every year. So you see the increase from 2021, and we are continuing this. To the left, you have the headquarters in Basel, in Basilea, where we are building also our new innovation space, and we are putting in place the Institute for Human Biology, which is headed by Matthias Ludorf. Of course, Hans Clevers is heading all of PRED, so that's to the left. And Hans Clevers is very well known in the space of organoids and ex vivo systems. So there are some brilliant TED Talk style presentations you can watch on YouTube or so with Hans. And then Matthias is also a very inspiring researcher to work with. On the Genentech side, we have Aviv Regev, who is heading Genentech Research and Early Development, GRED, and Aviv came from Broad. So she was one of the leaders at the Broad Institute, the Human Cell Atlas project, and many other things. And now she's heading research at Genentech. And she has recruited people like John Marioni from EBI EMBL, who is now helping to build out our computational efforts. I will come back to what this means. But let me just say very quickly, in Institut Roche, so why I felt that I needed to, I mean, I love Paris, so it was, but I also like California. It's a very nice place to live, etc. But when I got the opportunity to move back to Paris and to be part of Institut Roche, I took it because it's very close to my heart of what I've been working with for decades, actually, which is collaborative, multidisciplinary research. And this is what Institut Roche actually does. It's an independent, non-commercial research institute, but we are fully embedded in Roche. So all the people that work at Institut Roche, they are Roche. They have Roche badges, etc. But we are independent, non-commercial, and we work in the interface between our internal research teams. So PRED and GRED, Chugai, Foundation Medicine, Flatiron, all of these things where we operate. So these are companies that we own, and we work with external partners. So Roche is huge, over 100,000 employees, and of course we have a lot of interaction with the external world. So that could be direct. You come to me and you explain your research, and we say, oh yeah, of course, please do this, and we fund something where you have a specific task. You give us back some results that we want. That works. We do it all the time. Or we can partner. If you have a startup or you have some other ideas, then it's partnering that comes in. And we work with all these entities from the Institut Roche side. But we come in as the institute when it's really collaboration. So when we say that, oh, we have this fantastic idea of doing this and this and that, and we need both our internal research teams and external research teams. Then Institut Roche comes in and helps to maneuver the collaboration. So we are like an enzyme, a catalyst for others to actually perform together. And of course, I mean, the people that work at the Institute, they are 95% or more of them have a PhD, a postdoc. They are accomplished researchers in their own right. And we do contribute directly to research. But our mission is to help, to facilitate, to see to it that nice things happen. So let me talk a little bit about the vision we have and how this, let's say my world where I came from, mathematics, and the world of biology, how this could actually come together and help. This will be the thing, right? You all know Moore's law for computation and data. This slide illustrates that, you know, the computers are exponentially getting faster. We can exponentially store more data, and it's exponentially cheaper to do compute and store, right? So that should be wonderful. That should be good. At the same time, we have the reverse. So if you read more backwards, it becomes Aram. So Aram's law is basically saying that it becomes exponentially more expensive to develop medications or medical products. So actually, if you look at how we do development of medicines or drugs, right? It often goes, we start with cell lines, human cell lines. We try out thousands or millions of compounds on these cell lines, and then we come up with some ideas of the targets, things that we could target and molecules that we could use. And then we move it over to animal models. This is the traditional way of doing drug development. We move it over to animal models and try. Most things do not work then in vivo, in an animal model, but those things that do work, if they are really, really successful, we move them to other animals, and ultimately maybe some few things that pass through all of these, jump over all these hurdles, we move it into clinical trials and try it on humans, right? And more often than not, what works in a mouse does not work in a human, or work in a very different way in a human. So we need to do better. It's also very expensive. I mean, for every single product we put on the market, we have invested. It depends a lot on how you count, but you know, it's really expensive. Billions of dollars just for one drug. So we need to improve this. We need to do better. So what are we thinking about? Well, I mean, we are talking a lot internally within Roche about animal testing-free drug development. So we really want, I don't think it will happen very soon because also of regulatory issues. When you do clinical trials, etc., you need to have in vitro, in vivo, etc., proof that things work. But what we are thinking about is humanized cell systems. So really things that are more complex than cell lines. You need to have multiple different types of cells and be able, human cells, and be able to kind of make a biological avatar of what you think could represent a tissue or a human tissue, right? Try out things there in a little bit more complex scenario. At the same time, you build mathematical models. So let's say digital avatars that could mimic what you see in the biological avatars. On top of this, we get patient data. We get data from millions of patients around the world that we are treating with different things. All of this, so that's the ground truth. I mean, there is arguably no better model for human health and disease than a human, right? Human health and disease is the best model for human health and disease. So that's the ground truth. But if we can make all this connected and really come up with a vision of how to translate from one model to then another, we could advance much, much faster. And this was actually kind of the vision that convinced me to move from academia to industry. It was the vice president of cancer immunology at Genentech, Ira Mellman, when I was there interviewing for the position. He said to me that, Magnus, you know, if you really think that you can add even an epsilon, which is small, or an epsilon squared, which is even smaller than, right? If you think that you can do this, it's your moral obligation to come to us. Because we have the resources to move things like this. Yes, I discovered that that is true, but you know, 99% of the research and development happen outside of Roche. You are doing it, right? So this is also why I then moved to Institut Roche, because this is exactly what we do. We connect I don't think we even do 1% of the research within Roche. We do a lot, and there are fantastic researchers in our ecosystem. But most of the things are done outside, and so it's a very exciting interface to working. So I consider myself extremely lucky to be part of Institut. And then another thing is that, of course, traditionally, we have been working with data. Basically, often you come as a patient, you come in, the doctor takes blood tests or maybe a biopsy, and depending on what your condition is. And we have that data as a baseline. And then very seldom we do follow-up data sampling. But this is now what we are doing. We are pushing a lot that we should have. I mean, human health and disease is a dynamical thing, right? It's a very dynamical system. And everyone that worked in dynamical system knows that it's very hard to predict the dynamical system if you have one time point, right? You might be able to discriminate it from something else if you have a lot of prior knowledge. But if you have no prior knowledge, one time point tells you very little. So now we are starting to do longitudinal sampling. The vision we have is to detect the disease very early, before you even notice anything as a patient, and be able to push it back to a healthy position through different interventions before you even become sick. And we think many of the drugs that we currently have will work much better in an early setting. So this is another thing. And we do believe that we started talking about the human-machine partnership back in 2016, more or less, within Genentech. And this is how we think. We think that the ChatGPT, generative AI, all of the large language models, etc., they will help as an interface. They will connect us. Both, help us with some tasks where we humans are very slow. Our iPhone already help us with a lot of tasks, right? And the computer is in sequential things. It's so much faster than a human. So some things they do really, really well, but they can also help to connect us, biologists with computational scientists, etc. So this, we think, will be transformation. We can get this working. And the holy grail of our research is really to understand the immune system, because the immune system, human immune system, is always involved in health and disease. Whether you have, you're studying autoimmune disease or cancer, infectious disease, neurodegenerative disease, or you name it, we discover that the immune system is always there. And a lot of the drugs that we develop are actually immune modulating drugs. So we are pushing the immune system. It's a dynamical system. And we are, pushing it. It's a control theory problem in some sense for a mathematician. But of course, another thing that I discovered very early, decades ago when I started working with biology, is that we have very different language. So I still, when someone comes to me and tell me, I have a wonderful model for this, I don't think about that, right? This is what they have in mind, a mouse model. They have a wonderful mouse model. I think about this, right? Something like this, and I still do. That's the first reflex I have when someone says model. I don't think about an organoid, human, organoid system. I think about manifolds, equations, things that are moving. But these things will come together. This is what we should do in Uruguay, right? We should show them what we can do if we really work together as a community. Biologists, computational scientists, all of us contributing something together. So all models are wrong. And you heard this quote, I guess. Some are useful. This is Box, a famous statistician that said it. I never found that quote, but if you read this article and use ChatGPT, you can for sure extract it. I never heard him actually say that. But it's true, of course. All models are wrong, but some are useful. So you should always bear this in mind when you. But if we have many models, and all of them are connected and tell you the same thing, then it might be useful, even more useful. So we are thinking a lot in the kind of computational groups within WASH and Genentech about things like state space. So, you can imagine that you measure tons of things. You can measure DNA or RNA or protein, thousands or hundreds of thousands or millions of variables. So a lot of degrees of freedom that span something, because, when you do those measurements, of course, they span a huge space. If you measure 3 things, you can take every measurement and put it in a 3-dimensional space, right? If you measure millions of things, it's harder to imagine what that looks like, but you can still place the measurements in this million-dimensional space. And for a mathematician, we become so used to this, so we imagine that we actually can see it. Or am I wrong? You're right. I'm right. Yes, I think. So, but of course, it's a kind of hallucination, whatever, right? But still, a lot of the things that gives us intuition comes from thinking geometrically, right? And we think that we can see certain parts of this state space where we see healthy or unhealthy patients or physicians. And the goal of when we hit with a certain drug, this unhealthy state, it will move. And that, if we now measure longitudinally, we will be able to figure out, we hope, what trajectory that patient is actually on, and how we should intervene in order to push that patient back to a healthy position. And I think really this type of thinking has been around for a long time, for decades. It's systems biology. But a lot of people acted long before we had sufficient data. And maybe, I mean, now we have both much more data. We have longitudinal data, and we have things like large language models and generative AI that can actually help us to be an interface with that data. So what we are doing is not new in some way. I mean, if you went to the doctor 100 years ago, the doctor also measured things. Maybe took their pulse, felt, do you have a fever or not? Or, I mean, there were measurements, right? And then you come to a conclusion and you give some type of medication or not, right? The things will not change, but you might be, as a patient, we might be exposed to longitudinal sampling. You say, we do this, and you come back, and we see how things evolve, and we actually measure. And we do not measure only one or two or a handful of things. We measure millions of things and try to map out how you, as an individual, can be pushed back to a better position. And then, of course, I can't help. I mean, I love chess, and of course, in '97, when Kasparov lost against Deep Blue, that was a big thing. I mean, all over the world. And Kasparov himself, he has described that he became completely depressed. So, I mean, he felt that this machine had intuition and was, as a grandmaster. I mean, that's not supposed to happen. You know, I mean, a machine should not be able to outsmart the best chess player in the world, but it did. And it beat him, right? And he said, I mean, that's the end of it. I have nothing to live for, right? Of course, then he quickly realized that, yeah, we should team up with the machines, actually, and learn from the machines. And he organized, in 2005, there were this competition, where he teamed up with Deep Blue, or the new version of Deep Blue, and challenged the world, and he lost again. Right? There was a small team from, I think they were from the US, I have to check it, but there were 3 amateurs that actually connected their computers, their laptops, and worked together as a team, and that was much better. And then Kasparov said, oh, there is a law here. So super good human, super good machine, lousy process, that's worse than, weak human, weak machine, and a great process. Right? And I still think it's a good thing, right? We should create this very strong process of how we work together. Imagine a strong machine, strong human, strong process. I mean, nothing can stop us. This is at least how I think. Of course, I mean, when you look at machines, and I mean, there are a lot of dangers. I see them, but I think more about, I think we can do much better in terms of curing cancer, dementia, autoimmune disease. We can do much better. This is, I really feel this way. But we can only do it if we work together and truly, say that we will not give up. We will not be together for 1 month and think. We will be together until we solve it. 5 years, 10 years, right? but we will do it, right? And it's several of us think like this and come together. We can do it. So psychological implications. I heard a French philosopher on the radio the other day, and he was going like, "Oh, Kasparov, he went completely depressed. We will be completely depressed, all of us as scientists. We will have nothing to do. You know, the general artificial intelligence will be so much smarter than us." I don't think that will happen anytime soon. Every neuron in our brain is connected to 7 to 10,000 other neurons. If you look at an iPhone, it has less transistor, but it has on the order of as a cat. You take a cat, right? Then you have the same number of neurons in the cat brain as you have in an iPhone, right? But if you put the iPhone on the table here, and you put the cat on the table here, and then you wait to see if anything happens, you see the difference, right? The cat will do a lot of stuff when the iPhone will run out of battery, and then it's dead, right? And another thing is, of course, that a transistor in the iPhone is connected to 3 other things. There are 3 ports for a transistor. The neuron in the cat brain is connected to 7 or 10,000 other neurons and a lot of other cells that feeds information. So imagine the space that we can span. I mean, for each neuron, I mean, it would be 2 to the power of 7,000 or something like that, potential future states, compared to 2 to the power of 3, which is 8. It's a huge difference. So I think that we will be, let's say that that's intuition, maybe, right? So we will still be artists, and we will for sure be decision makers. We will make the calls. The machine will help us with a lot of things, but we will make the calls. Let me switch gears and try to wrap up with some examples. So cancer is maybe the best example that we have. Over the last 10 years, we have transformed cancer care. So I will finish with this example. Before we could cut, we've been cutting for thousands of years. I mean, the Egyptians, they cut away tumors already, 4,000 years. And then we discovered that we can burn things with radiation. We can poison with the chemotherapy, and that has been the standard of care for a long time. But then, 2018, the Nobel Prize was awarded for checkpoint inhibitors. So monoclonal antibodies that target a checkpoint on T cells, so immune cells that attack tumor cells. And since over the last 10 years, we have advanced a lot in terms of immunotherapy, and that is actually stimulating the immune system to do its work. That's the idea. Cancer is very complex. There are always mutations. Some cancers have more mutations than others, so it's hard to read here, but it's colorectal, lung, and melanoma. They have a lot of mutations in these types of tumors, and that's natural. I mean, we eat a lot of different strange things. Our skin is exposed to a lot of strange things, and our lungs. So highly mutagenic things, and often our immune modulating treatments, they work better if it's highly mutated, which makes sense because these tumor cells look more different compared to a normal cell. So it's easier for the immune system to recognize them and hit them. These are cytotoxic T cells attacking a tumor cell in the middle. We have a lot of imaging. This is, of course, in vitro. It's just cells. But we also start to have a lot of in vivo imaging of how cells interact and work together. Conceptual models are important. So let me just walk you quickly through how we think that, let's say, solid tumors work, right? So you have these mutated cells, cancer cells, And since they are mutated, and that translates ultimately into proteins that look a little bit strange compared to normal, and they are shut up, they are packed up in small pieces that we call peptides that look slightly different, and they are then recognized by immune cells like dendritic cells. They are picked up, they are brought to a lymph node somewhere in the body, and there, T cells are waiting and recognize, oh, this is a peptide that I recognize. If they do it, they clonally expand. They make millions of copies of themselves, monoclonal expansion. And they then travel through blood to the tumor, infiltrate the tumor, and kill the tumor cells. And this actually goes on all the time in our bodies. Our immune system is protecting us against cancer by these steps. But sometimes one of these steps fail for some reason. And then it's our job then to try to figure out why. And if we can help the immune system to overcome that hurdle and actually make the immune system go round. And this cures people. So, I mean, if you look at melanoma, for instance, people that 10 years ago were terminally ill, they had weeks, months, or weeks to live, are now getting checkpoint inhibition injected every 3 weeks, and they are cured. They're completely cured. The cancer disappears, and they are cured and can live a long and healthy life. But it only works for a small proportion of the patients. So in the best of cases, maybe up to 50%. Normally, rather 20 or 30% of the patients respond. And in some cancers, much less. And we don't know why. So this is, again, a huge challenge for mathematicians. We have a system, we perturb it, and some initial states lead to recovery. And some other initial states, they don't move, or they become worse, which is even worse. So this we need to work on. So mathematically, again, we think about this, everything we measure is in some type of state. The cancer immunity state space, I will skip rapidly because I want to focus on a few other things before we close. Is that okay? You're still with me? Yes. So one thing that we have realized is that in many diseases, we have a low-grade chronic inflammation. You see it in cancer, you see it in autoimmune disease, you see it in neurodegenerative disease. So this type of chronic, low-grade inflammation is not good. So you are, from a dynamical system point of view, you have this healthy thing, the sun goes up and down, we have the circadian rhythm, and everything is fine. All of a sudden, something hits us. Could be an infection, right? And we are thrown off. If everything works well, we are pushed back to this healthy homeostasis, this healthy position. But sometimes we get stuck in another chronic, low-grade inflammatory state, and we develop cancer or autoimmune disease or dementia, many other things. And then we need to push them back. So let me go very quickly through what this can look like, because it has been very abstract. 5-minute run through on one of our large phase 2 clinical trials in bladder cancer, metastatic bladder cancer. So the data here is, all of this is open, you can download it, you can play with it, etc. And we are actually sharing a lot of data globally with the research community. So this is a 3D PCA of samples. So every little point here is a patient. They are colored according to how they respond. So we do imaging and look at the tumors and see if they shrink or grow. The light green are complete response. Then you have partial response, stable disease, and progressive disease. You cannot see anything. So I just took something that is sitting in 25,000 dimensions and projected it down to 3D the best I could to keep as much of the information as possible, I don't see much. So you need to do better. So here I said that, okay, if they are close together, they might be close together also from a biological point of view. But thinking about a manifold, a mathematical manifold or something like this, I mean, the Euclidean distance, the straight line between, if you imagine this space where they are lying, right? The straight line will not be the line that biology has to travel. Biology has to follow the rules, like following this strange space to go back somewhere. So what I did was that I connected the nearest neighbors, I built a graph, and I said that distance must follow this graph. You must walk along this graph in order to come from one point to another. And then I used that as a global distance for capturing as much as possible. And now maybe you can start to see that some areas have more red and some have more green. And also there is a taxonomy in bladder cancer that we use that happens to come from my home university, Lund. So it's called the Lund taxonomy. It's used all around the world for bladder. And you see that a lot of the areas we see here correspond to their classification. It's not too far away. If we now move, so the subtyping of Lund is based on immunohistochemistry and a lot of other things, so much more than what we looked at here, where we look at only. What I did next is that I looked for what is more relevant for actually going from complete response to progressive disease. I mean, if I do what we call rank regression, which basically finds me the genes that mostly discriminate between those different states, the best discriminating genes. And then I plotted the genes to the right, the patients to the left. And if you look at the green, you see the genes pointing in the same direction. They are upregulated. So a lot of them has to do with cell cycle, and then some of them have to do with, let's say, inflammatory signaling. So again, coming back to this inflammatory state that we always see in cancer. Others have of course seen this also. I mean, this is not a new thing that we discovered. I mean, again, a kind of graph-based plot where you clearly now see some areas here that would have more green than red. So then we can start to map out what are those areas. It's kind of one way to look at the cancer immunity state space. These areas are better. This is a paper by Kevin Litchfield and from Charlie Swanton's team. I think actually Kevin is coming here in March to Montevideo and Uruguay, so working with us. But in any case, they have found the same thing across huge collection of solid tumors. So there is something here. Another thing is then when I started to look at what is in the kind of orthogonal components compared to this inflammatory response, then I found another signature, which is the ApoBec signature. And ApoBec is actually proteins that act on mRNA and edit mRNA, so they can put in like softly put in mutations. ApoBec has been observed in other areas. You do better if you're inflammatory high, CXCL9 high. You do better as a patient, but you still see that this is a survival curve, and you still see that if you have a low inflammatory state, you do worse. If you have a higher inflammatory state, you do better. ApoBec should also be high, and if you're both ApoBec high and CXCL9 high, you have an almost 60% survival, which is much better than the normal survival in metastatic cancer. So what we should do together is to learn more about this. And we are actually, I think I'm coming to this now, so we are actually organizing some events. So I finished in 3 minutes, no worries. But we are organizing some events. We call them research camps, and where we actually bring both data and questions that we are working with external researchers as well as internal. So we bring people from Genentech, Roche, but we also invite other people to come and work with us. This is a few examples of research camps that we've had before. The left there is in Zurich in 2021. I think you recognize Arthur there on the side. There, Arthur Avila, a Fields medalist that has been to actually the camps we've made so far. A world-leading expert, of course, in dynamical systems. But there are a lot of really distinguished scientists that come to these events and work together. Now, these are sprints. It's 1 week. Let me see if I have a presentation of the camp we are running in Uruguay. So it's a sprint, right? It's a 1 week exercise. So we get together. We will do it the 4th to the 9th of March in Punta del Este, actually, Solanas Convention Center, which is a good location because it's very hard to escape from there. So if you're there, you maybe don't want to escape. It's a very nice place to be. And when we have this, you definitely don't want to escape because you swim in science and research 24 hours per day. You have to sleep sometimes, but you do it as short as you can, right? Because it's really, you have colleagues from all different areas working together, and we bring data, we bring questions, and then we hands-on attack those questions. So there will be, at this camp, there will be questions around cancer. We are bringing a lot of clinical trial data. There will be, let's say, neuro disease. We are bringing a huge autism data set that we are very excited about. But there are also a lot of partners to us that are coming in and working with us on the interface. So we are running a lot of our internal platforms around collaborative computational science, where biologists and mathematicians will be able to interact together on the same data and on the same questions. And there will be a lot of other external companies, not Roche, but other companies that we work with to kind of build this infrastructure for us. And I think, I think with this, I stop, and I'm super happy to take any questions. Muchas gracias. Sí.
Esta mañana tuve un evento que se hablaba de innovación social.
Sí.
Y la importancia de colaborar las grandes empresas con las startups y aportar algo nuevo entre el elefante y la gacela, ¿no?
Sí.
La capacidad que tiene la startup de ser más ágil y la gran empresa de proveer todo eso que la pequeña. Tú mencionaste eso al respecto, de mucho trabajo colaborativo. ¿Podés profundizar un poquito?
Sí, lo que pienso, claro, nosotros tenemos miles de investigadores internos en Roche, Genentech, etcétera, que están trabajando, pero muchas veces con una meta bastante bien definida. Y yo creo que muchas veces lo externo puede darnos empujar un poquito en una dirección que no estamos siguiendo nosotros mismos. Que quizás es un camino más fácil a llegar a lo que tenemos que hacer, a lograr hacerlo juntos, ¿no? Eso es una cosa. Después, claro, cuando cambié del mundo académico al mundo de la industria, yo creía que el cambio iba a ser brutal, grande, y después, nada, era lo mismo. ¿Viste? La gente, los investigadores son siempre investigadores. Ellos tienen, vos como persona, vos tenés tu curiosidad, quieres ir de un lado al otro, y eso es lo que empujas. Y es lo mismo si vas a Genentech o Roche, va a ser lo mismo. Claro, alguien te va a decir que tenés que pensar en cáncer. Eso es importante. Y vos empezás a pensar y después ves un camino y empezás a caminar ese camino. Es lo que hacemos todos, ¿no? Pero después eso de realmente escuchar, a vos tenés una idea de cómo uno podría mejorar una cosa, bueno, lo probamos juntos. De innovación, eh, yo creo que la innovación se crea en los interfaces. Cuando salís de tu campo y encontrás otras cosas, eso me imagino todos los startups que tienen aquí en Montevideo, Uruguay, por ejemplo, han sido creídos de esa forma. Que alguien está pensando discutiendo con un amigo, saliendo, oh, eso sería genial, y uno empieza a probar. Y bueno, nosotros queremos crear esas situaciones, más o menos.
¿Y cómo lo tienen institucionalizado el espacio para facilitar?
Bueno, en realidad, claro, mi instituto...
Hay que innovar y todavía crear.
No, pero mi instituto es lo que hacemos. Nosotros tenemos esa misión de Roche. Estamos y claramente tenemos un montón de colaboraciones alrededor del mundo que estamos manejando en plataformas internas, digitales, por supuesto, pero a veces yo creo que ese contacto humano, ¿viste? Humana quizás se dice, ¿no? De encontrarse, de estar juntos, de tomar el café de la mañana, discutir, y es también muy importante. Es por eso que hacemos esos eventos, los research camps, para estar juntos y hablar y tener somos seres humanos, no somos máquinas. Tenemos que vivir la vida, tenemos que compartir la vida, tenemos que estar juntos. De esa manera pueden pasar cosas lindas.
Esta idea o esta aproximación de trabajo, de Research Camp, ¿es un invento del instituto o esa idea ya existe?
No, no es un invento del instituto. Yo aprendí eso trabajando con the European Consortium for Mathematics and Industry. Empecé hace 30 y más años, 25 años, trabajar con esa. Es la organización más grande de Europa de matemáticas aplicadas. Y ahí hemos hecho study groups with industry durante 40 años, algo así. Y claro, funciona más o menos de esa manera. La industria viene con una pregunta, nosotros armamos un equipo de matemáticos que tienen que atacar ese problema, pero después empezamos a desarrollar eso, yo junto con amigos. Cuando yo estuve en Pasteur, en el Instituto Pasteur, hicimos un research camp aquí en Punta del Este 2015, que se llamaba MIST Camp, Modeling Infectious Disease and Pathogens Interaction.
Pero con este mismo concepto de tomar el problema, brainstorming, así.
El mismo concepto, tenemos un video que vos puedes encontrar por YouTube, te puedo pasar así, y te paso el trailer. Aquí también vamos a tener un equipo de filmación que va a grabar todo. Y ese equipo que hacen la producción de material forman parte de una experiencia, bueno, creativa. Queremos grabar y queremos poder compartir cómo se puede colaborar, de cómo se puede desarrollar juntos. Y yo realmente creo que tenemos una ventaja aquí en Uruguay, porque nos conocemos, es pequeño, como yo dije de mi pueblo, es pequeño, todo el mundo se conoce y ahí puedes encontrar las personas que realmente tienen ganas de hacer algo juntos y de hacerlo no solamente una semana, pero continuar a trabajar juntos y solucionar cosas. No, yo tengo fe, yo creo que Uruguay es ideal. Además es una entrada para Latinoamérica, para muchas cosas, creo. Es un punto estable donde puedes desarrollar cosas y después transmitirlo a toda Latinoamérica y el mundo después.
Hola Magnus.
Sí, hola.
Mucho gusto, muy buena la charla. Quería preguntarte una cosa, en todos esos institutos que tú has estado y organizaciones, ¿alguna vez estudiaron las técnicas orientales de medicina en forma científica, más científica, así como más matemática? Como por ejemplo la acupuntura, por ejemplo.
Sí, claro, lo que pasa es que en el Instituto Roche, ¿viste? La única, te digo, las únicas fronteras que tenemos, las únicas restricciones que tenemos es que tiene que ser interesante para nuestros equipos de investigación interna. Eso significa que, claro, Roche es grande, tiene diagnóstica, tiene farma, entonces estamos trabajando en prácticamente todas las enfermedades y también tenemos esa parte de diagnóstica. Eso significa que casi todo que uno puede imaginarse de hacer en biomedicina lo podemos ayudar, podemos generar, podemos estar ahí. Hasta también la inteligencia artificial, el desarrollo de plataformas, modelos matemáticos que sirven exactamente para eso. Claro, no estamos trabajando en áreas que no está en el portafolio de Roche. Ahí hay una limitación.
La pregunta es, diferente del Research Camp, ¿cuáles son las formas de interaccionar con el instituto?
Bueno, sí, muy buena pregunta. En realidad, cada investigador en el mundo puede contactarnos. Tenemos esa página web, instituto.roche.com, etc. Y ahí, o googleás Instituto Roche y ahí estamos. Y también tenemos una interfase donde puedes escribir y tomar contacto con nosotros. Y nosotros trabajamos directamente con nuestros equipos de investigación, pero también con partnering y todas las otras partes del ecosistema Roche. Pero eso, el Research Camp realmente es solamente pequeño, pero es un vehículo que usamos para crear contactos, para extender la red que tenemos y que ustedes todos tienen.
Si hay más preguntas, capaz que puedo participar. Y creo que igual se puede dar diálogo
Obvio.
informal, que algunos capaz que se anima más a conversar o preguntar en el uno a uno.
Perfecto. Bueno, fue muchísimas gracias de escuchar y bueno, seguimos charlando, así, seguimos en contacto. Muchas gracias.